5 minutes.
Oh, okay, ma'am. Okay.
Okay. Am I audible, Anu?
Yeah, ma'am.
She'll be joining in 5 minutes.
Okay, okay.
We'll be starting the CME in another 5 minutes. Sorry for the inconvenience.
Okay.
Hello, good evening.
Good evening to one and all. Welcome to our National CME Srishti on the topic genetics and embryology. Today we will build on the foundations laid day before yesterday and dive deeper into the topic embryology. Our expert faculty will share their insights and experiences about the topic. It's my honor to introduce our resourceful speaker, Dr. Geetha Jayachandran, a distinguished expert with an impressive 24 years of experience in teaching and in the field of embryology. She currently works as an associate professor in Department of Anatomy, Paripally Medical College, Kollam. We are fortunate to have Dr. Geetha leading a comprehensive six-part series on the topic embryology, with today marking the second session. We are eagerly to hear your session, ma'am. Without further ado, I welcome you, ma'am, to begin today's scientific session. Over to you, ma'am.
Can you hear me?
Yeah, ma'am.
Oh, ma'am.
Audible,
Sure.
audible.
Okay, can you see the screen also?
Yeah, ma'am. We can see your slides. Could you please put in the slideshow, ma'am?
No, my laptop is not allowing the slideshow. I
Okay,
will
ma'am.
enlarge the screen. It's a problem with my laptop. Okay.
Okay, it's in.
I will do it once more. Okay.
Okay.
Ma'am, if you can share the entire screen, then maybe it is possible.
Let me see once. Some sort of error is showing. You're presenting and not annotating. What means what?
I'm actually you're meeting co-host now. That's why you're not taking.
What's it?
Uh, we are make you as a co-host. That's why.
കോഹോസ്റ്റ് ആക്കിയതുകൊണ്ട് കുഴപ്പമൊന്നുമില്ല.
Uh,
എന്നാലല്ലേ ഷെയർ ചെയ്യാൻ പറ്റുള്ളൂ. ഇതിപ്പോ സ്ലൈഡ് വ്യൂ പറ്റുന്നില്ല മാഡം.
സ്ലൈഡ് പവർ പോയിന്റ് കാണാമോ?
കാണാം കാണാം.
പക്ഷെ എനിക്കത് മൂവ് ചെയ്യാൻ പറ്റുന്നില്ല.
ആണോ?
നെക്സ്റ്റ് സ്ലൈഡിലോട്ട് ആക്കാൻ പറ്റുന്നില്ല. എനിക്കത് ബ്ലഡ് ടു അവോയ്ഡ് ആൻ ഇൻഫിനിറ്റി മിറർ ഡോണ്ട് ഷെയർ യുവർ എൻറ്റയർ സ്ക്രീൻ ഓർ ബ്രൗസർ വിൻഡോ. ഷെയർ ജസ്റ്റ് എ ടാബ് ഓർ ഡിഫറെൻറ്റ് വിൻഡോ ഇൻസ്റ്റഡ്. അങ്ങനെ വരുന്നു. ഒരു മിനിറ്റ്.
ആണോ?
ഒരു മിനിറ്റ്. Can you see it now?
Yeah, ma'am, slide is visible. Even in this size also, it's okay. Can you move the slides?
Is it okay? Is it moving?
No, it's not moving. It's in the same second slide itself.
Okay, now is it?
Now it's moving. Ma'am.
It's
Now it's
moving,
moving.
But it is not in the slideshow, okay?
Yes.
I think there is
That is
some
okay,
problem.
ma'am.
Okay. So we'll start.
Okay.
We'll just try once more. So
No,
can you
ma'am,
see
it's
the
okay.
slide? Okay. Now this is the recap where we have seen the cleavage that occurring in the fallopian tube, and at the Morla stage it has reached the uterine end of the fallopian tube, and in the early blastocyst stage with the zona pellucida it is entering in the uterine cavity, and with the disappearance of zona pellucida by the hatching process it starts to implant, and during the first week the starting of implantation starts but to end in the the second week. So this is the blastome here with the zona pellucida. The next picture, the early blastocyst with zona pellucida, and the second figure is the late blastocyst without the zona pellucida, and that is the hatching process. Okay, so we have also seen that with the implantation, the trophoblast, the outer cell layer, has started eroding into endometrium, and they have started to differentiate into two. Only little bit started to invade the endometrium, and the trophoblast is multiplying to form two layers, the cytotrophoblast and the syncytiotrophoblast. Is it the slide moving? Hello?
Yes, ma'am.
Okay. Now, this was the stage where we ended last week. You can see the blastocyst getting attached, and the cytotrophoblast, which is the cellular layer, getting eroding into it and multiplying, and you can see that cell nature is getting lost as it goes deeper. And in the next slide, that is, we have finished the second week, and then next we are coming to the first week, we are going to the second week. So from second week, during the second week and third week, what we say is we will concentrate on each part, that is the trophoblastic part and the inner embryoblast. Okay, first coming to the trophoblast. So the same picture, that green area, you can see a light blue cellular, that is the cytotrophoblast. Now concentrate only on the outer cell layer that is converting into cytotrophoblast and the syncytiotrophoblast. We will first finish that and then come to the embryoblast. Okay. And so the cytotrophoblast, that is multiplying rapidly, eroding into the endometrium, which is full of engrossed blood vessels and glands. The cytotrophoblast, as it multiplies, it will erode into the endometrium deeper and deeper. So the same thing, it has evolved some more, and you can see that the cytotrophoblast is covering the entire developing embryo, but it is more developed at the embryonic point. And the syncytiotrophoblast also, it is more, even though it is going deep, more rapidly multiplying, it is more on the embryonic point. Because of it dividing so rapidly, so many cavities appear in the syncytiotrophoblast. So these cavities are called the lacunar space. Okay, And in this figure, you can see that the lining endometrium, and this is the site of entry of the embryo into the endometrium, and it is initially filled with fibrin clot. Okay, now what is the stage of this syncytial trophoblast? It is so much rapidly dividing. You have so many cavities in it, and that is the lacunar spaces. So in the next picture, this is the same picture, but here you can see that the rapidly, excuse me. Okay, the rapidly multiplying syncytial trophoblast is eroding into the nearby blood vessels. So what will happen? The blood will enter the lacunae. Okay. In the previous picture, I forgot to mention one thing. So you have lacunae, and in between you have syncytial trophoblast. Got it? That is called the trabeculae. The trabeculae is the syncytial trophoblast that is present between the lacunae. So
and the slides are not moving.
Is it moving now?
Ma'am, actually, we can see your second slide.
Now it's moving, ma'am.
Okay. I will do it. Have seen this?
Yeah, ma'am.
Yeah, ma'am.
This you have seen?
Yeah. No, that we haven't seen. Now it's visible.
Okay. So today's topic is actually the second week and third week. And in the second week, with the implantation starting, we have started with the implantation last week, so it has to be completed in the second week. And so during implantation starting, you have the inner cell mass and outer cell layer, which is the trophoblast, which is eroding and forming the syncytial trophoblast. Okay. And when we talk about the second week, we will be first dealing with the trophoblast, that is the outer cell layer, and later we will coming to the inner cell mass, which is the embryoblast. So first coming to the trophoblast, that is the outer cell layer that is going deeper and deeper into the endometrium. Can you see the cursor also? Can you see the cursor moving?
Yes,
ma'am.
Yes,
Okay.
ma'am.
So in this picture, you can see that the blastocyst is entering into the deeper and deeper, and the cytotrophoblast is multiplying more on the embryonic part. But in the next picture, you can see that it's almost fully inside, and it is almost covered with the syncytial trophoblast also, but it is more in the embryonic part. Because the syncytial trophoblast is rapidly dividing, that is why it doesn't have the cell membrane, and it is called syncytial trophoblast. Because of the rapid multiplication, you have lot of spaces in it, and that spaces are called lacunar spaces. And when so many lacunar spaces are developed, it will have some pillars of syncytial trophoblast in between. Getting it? The syncytial trophoblast, you have so many spaces, and in the space between the spaces you have these syncytial trophoblast pillars. That pillars are called trabeculae. And when you imagine the developing embryo, think it as a sphere thing, and the whole thing is covered with a syncytial trophoblast, and the developing trabeculae and the developing lacunae is everywhere, but more on the embryonic part. So one lacunae can get it will be extending into the next lacunae. So you can think of the loaf of bread in which the ants will enter, and it will be coming out through another opening in the loaf, isn't it? So it will be a network. This trabeculae will finally form a network, and in between you have the trabeculae. And as a result of rapid multiplication, you have first the lacunae. Later, what will happen is it will erode into the surrounding blood vessels, both vein and arteries. The spiral arteries in the endometrium filled with blood will be eroded by the syncytial trophoblast, and this will, the maternal blood will enter into the lacunae. So that is the starting of the uteroplacental circulation. Now, when you look into the cytotrophoblast, which is inner to the syncytial trophoblast, next picture, when you look into the cytotrophoblast, look into the cytotrophoblast alone, here you can see it is a continuous layer. But here you can see there are finger-like projections. And where are they going? They are going into the syncytial trophoblast. Getting it? Even though the cytotrophoblast, initially the cytotrophoblast has given rise to syncytial trophoblast, with the appearance of lacunae and the trabeculae, after the formation of trabeculae, the cytotrophoblast will extend into the trabeculae, and this is called the primary villi. You can see the primary villi, and with the appearance of the primary villi, the lacunae filled with the blood is converts its name into intervillous space. Hope it is clear. That is the development seen in the second week related to the trophoblast. Now coming to the embryoblast. Now, here in this picture, you can see that the embryoblast that was attached to one pole is getting organized to form layers. Okay, and the layers, the first layer that is formed is the lower one, that is the hypoblast. That get organized to form cuboidal cells. You can see cuboidal cells here getting or forming a layer. That is the first layer to be formed. That is hypoblast. Epi means to the surface, epidermis to the surface. Hypo means below that. So you have hypoblast or primary ectoderm, which is the first layer that is formed. Later, what will happen? The other cells that is grouped above will get a cavity. And as a result, the cells will be forming columnar cells on the dorsal aspect of the hypoblast, that is called the epiblast or the primary ectoderm. What I have written is wrong. Epiblast is ectoderm, primary. What I wrote it is wrong. Hypoblast is endoderm. Epiblast is ectoderm. Okay, and a cavity is formed that is called the amniotic cavity. So when you see about the amniotic cavity, you can see some cells that forms the roof. Those cells are called amniogenic cells. Amniogenic cells. So there is a second thought of school of thinking that this amniogenic cells are derived from the trophoblast. Previously I said there is a cavity that appear within the embryoblast. That is one school of thought. Another is the amniogenic cells are derived from the trophoblast. So the amniotic cavity, when we talk about the amniotic cavity, it is lined above by the amniogenic cells and below by the primitive ectoderm or the epiblast. Okay. So now think of the embryoblast in the globular structure. there will be a circular disc of tissue. The embryoblast is arranged as a circular disc that lies in the within the trophoblast as a circular disc with epiblast above and hypoblast below. So the dorsal surface of the embryonic disc is the epiblast and the ventral surface is the hypoblast. Okay. Now, in this diagram, here in the periphery, and if you think of that circular disc, in the periphery you will have there is a continuation of the embryonic disc with the structures and the periphery. Now we have seen that the embryonic bilamellar germ disc is formed, and the bilamellar germ disc consists of epiblast, a basement membrane, and the hypoblast. Now, this was the blastocele, isn't it? The blastocele, but an extra lining has come here. That is called the Husser's membrane. And the Husser's membrane, again, there are two schools of thought. One is it is derived from the hypoblast, the migrating cell, or it is the layer that is formed from the cytotrophoblast. And with the appearance of the Husser's membrane, the blastocyst cavity is now called the primary yolk sac. Okay, and at the periphery of the embryonic disc, you have an amnioectodermal junction. Can you imagine that? Can you get it? I will say once more. The embryonic disc with the formation of amniotic cavity, it is a disc, circular disc with amniotic cavity on one side and yolk sac on the other side, and the disc is attached to the cytotrophoblastic layer along its periphery of the circular disc. Getting it? Hope you are getting it.
Yeah, ma'am, it's clear.
Now, this was the previous picture which I have shown, but here what happens is this layer, the Husser's membrane, has become thin, flattened cells. Okay, and there is an extra layer that is developing between the Husser's membrane and the cytotrophoblast. That is called the extraembryonic mesoderm. Extraembryonic mesoderm or primary mesoderm. Extraembryonic mesoderm doesn't have anything to do with the embryo proper. It is seen outside the embryo. So that is extraembryonic mesoderm that is developed between the yolk sac and the cytotrophoblast. And small cavities develop in this extraembryonic mesoderm, splitting the extraembryonic mesoderm into two layers. One goes with the yolk sac, one goes with the cytotrophoblast. And that cavity extends all around except one region that forms the connecting stalk. So because of the division of the extraembryonic mesoderm that goes with the cytotrophoblast and that goes along the amniotic cavity, it is called the somatopleuric mesoderm, and that goes with the yolk sac along, it is called the splangnopleuric mesoderm. Okay. So as a result, the cavity expands and the primary yolk sac is now this is that extraembryonic cavity, and as this cavity enlarges, it pinch off the major portion of the primary yolk sac. You can see that primary yolk sac is large, but in the second picture, the yolk sac is reduced, and you can see a lot of this yolk sac, part of the yolk sac is pinched off at the expansion of the extraembryonic coelom, and that pinched off area, it is called the extracoelomic cyst. And the yolk sac, which has reduced in size, is now converted to a secondary yolk sac. Now, what is amnion? Amnion is this amniogenic cells, and which the extraembryonic mesoderm here, which is it is not splagnopluric, it is somatopluric mesoderm. So amnion is the amniogenic cells with the somatopluric mesoderm, because this splagnopluric is only related to the yolk sac. And what is chorion? Chorion is this extraembryonic mesoderm, that is the somatopluric mesoderm and the trophoblast, syncytial trophoblast, and the cytotrophoblast, that forms the chorion. So that here you have the primary villi inside, so that is the chorionic villi. That is why it is called chorionic villi. So I have said that the embryo now it is circular, with on dorsal aspect you have the primary ectoderm, and on the ventral aspect you have the primary or primitive endoderm, and this is the circular embryonic disc. And the dorsal ectoderm, the primary ectoderm of epiblast is columnar, whereas the hypoblast is cuboidal cells. But in one area, there is a circular area in the hypoblast which is columnar. That area, it is called the procaudal plate. It is called the procaudal plate. The appearance of procaudal plate indicates the axis of the embryo. So with the appearance of the procaudal plate, you will be able to know the head caudal axis, craniocaudal axis. Just this procaudal plate is the site for the future mouth. Now, so we have seen so many events. So this second week is called the week of 2. Why is it called week of 2? Because you have the bilamellar germ disc, which is the hypoblast or primitive or primary endoderm, and epiblast, it is the primary ectoderm. You have two cavities. The dorsal cavity, that is the amniotic cavity. The ventral cavity is the yolk sac. Again, the yolk sac, you have primary and the secondary. The extraembryonic mesoderm is also divided by the extraembryonic coelom into somatopluric mesoderm and splagnopluric mesoderm. So many events are happening in the second week, which is that is why it's called the week of 2. Now in this picture, you can actually see that this is a big balloon, and inside that you can see another balloon, and inside that you can see 2 balloons, and the 2 balloons are between that you have the area of contact. Inside one balloon you have 2 balloons, and that 2 balloons are compressed so that you have a disc in between. Now coming to the third week, again we are going to talk about the trophoblast first, and then the embryoblast. So now we have seen that there is primary villi, primary villi is formed by the cytotrophoblast getting into the trabeculae. Trabeculae is formed by syncytial trophoblast. So when you take a cut section and see, in the middle part you will have cytotrophoblast, and on its periphery you will have syncytial trophoblast. Now, this is the mesoderm, the somatopleuric mesoderm. In some areas, you can see that it extends into the core of the cytotrophoblast. So that is that if you have the entry into the cytotrophoblast, then it becomes the secondary villi. Primary villi, you have syncytial trophoblast, outer, and a core of cytotrophoblast. When it comes to secondary villi, it is syncytial trophoblast outer, middle layer of cytotrophoblast, and a core of extraembryonic mesoderm, that is somatopleuric mesoderm. So that is secondary villi. In this picture, what you see is the dark green is the syncytial trophoblast, and inside that you can see the cytotrophoblast, and inside that you can see the whitish area that is actually the extraembryonic mesoderm, and in that you can see the blood vessels arising in the mesoderm. That is the appearance of blood vessels, that is the tertiary villi. Tertiary villi. So tertiary villi, when you take a cut section, what all things you will have? You will have syncytial trophoblast out, then inner to that you have the cytotrophoblast, inner to that you have the extraembryonic somatopleuric mesoderm, in that you have the blood vessels. So, okay. Now, at the last stage of the third week, you can see from the tip of the primary villi. This primary villi doesn't allow all the other components to get through and through. It will be limited to some thickness. Okay, the tip of the primary villi will be joined with the tip of the other villi along the outer aspect of the intervillar space. Okay, and that forms a shell called as outer cytotrophoblast shell. So the tip of the villi doesn't allow the other components to pass through, but the tip joins together through the periphery and it forms a shell that is called the outer cytotrophoblast shell. So this is the cross section which I was telling. The primary villi, secondary villi, and tertiary villi. The primary villi cross section, in the inner part you have cytotrophoblast and syncytial trophoblast. Then you have the mesoderm, extraembryonic mesoderm, cytotrophoblast, and syncytial trophoblast. Here you can see the in the syncytial trophoblast you have see that the blood vessels, then cytotrophoblast and syncytial trophoblast. Now the villi has formed. Now only the maturation of this villi will develop. So implantation, primary villi, secondary villi, tertiary villi, and the outer cytotrophoblast shell that has contributed to the development of the pre-runner of the placenta. Now coming to the embryoblast. Changes in the embryoblast. Now we have in the first week, by the end of the first week, the embryonic disc is circular. The feature that leads to the that leads us to find out which is the cranial end is only the procaudal plate. So the in the third picture you can see that this is you are seeing from the ectoderm, from the dorsal aspect, the primitive ectoderm, that is the epiblast. In that the cranial end is the procaudal plate or the precaudal plate, and the caudal end, you have a thickening developing in the epiblast. That is the primitive streak. How is primitive streak formed? The ectodermal cells move towards the cranial caudal end near the midline. So you can see that as this migration of cells into the caudal region occurs, the rounded shape of the embryonic disc is changed to oval. So this primitive streak formation has contributed a little bit to the change in the shape of the embryonic disc. So coming to the details of the primitive streak. So this is the cranial end. So when you talk about the embryo, we say it has cranial and caudal and dorsal surface and ventral surface. So this is the area where you have the procaudal plate, that is a feature that is seen in the primitive endoderm or the hypoblast. This primitive streak is in the primitive ectoderm or the epiblast. Okay, so initially there is a keeping up of the primitive ectodermal cells into the midline in the caudal region of the disc, and that elevation has a cranial elevated area known as the primitive node with a pit in it. That is called the primitive pit, and it is also known by the name Henson's node. And this pit is running along caudally as a groove. That is the primitive groove. So you have on the dorsal aspect a primitive pit with a caudal extension, that is the primitive groove. So primitive node, primitive pit and primitive groove. And it is considered as the primary organizer. Primary organizer means the formation of primitive streak induces another structure to be formed. That or structure will induce another structure to be formed. So into the next slide. So you can see the primitive streak, primitive node. This is epiblast. Now the main event that occurs in the third week is called gastrulation. Gastrulation means the conversion of the bilamellar germ disc to trilamellar germ disc. This is the epiblast. So this you can imagine the cells from the sides migrate into medially and then dips into the under to the primitive epiblast. The cells from the side migrate towards the primitive streak, and into the groove it goes and occupies the space between it. Okay, that's the movement of the cell. So gastrulation is the process by which the three layers are formed. So when the primitive cells from the primitive streak, first what will they do is they display certain cells in the hypoblast, and with the displacement of the cell, they change their name to the from primary to the definitive endoderm. So the hypoblast or the primitive endoderm is now becoming definitive endoderm, and some cells will migrate to occupy space between the epiblast and now the definitive endoderm, and that cells occupy all of the area of the embryonic disc except certain regions. So this will form the mesoderm. With the development of the mesoderm, the epiblast will be converted to definitive ectoderm. That is the primitive ectoderm will be converted to definitive ectoderm. So this is the inner cell mass, the amniotic cavity. Then you can see the how the primitive streak and the cells are getting in between these two, and that is the process of gastrulation. So which all areas? So this is the primitive pit, and you can see the cells migrating, and this is the area of procaudal plate, and this is the primitive streak area. This is different, and you can see some cells are spreading in the side. That is the formation of the intraembryonic mesoderm. So we have mentioned about the extraembryonic mesoderm before, but that doesn't contribute to the embryo formation. This is the intraembryonic mesoderm. Okay. So what will happen to the primitive streak? The primitive streak actually was in the caudal region, but as it developed, you can see that the rounded or the more rounded embryonic disc is becoming more and more elongated and spear shaped. Okay, and by about 26th day, the primitive streak regresses, and you can see that this is the ectoderm that is seen. The ectoderm, with the primitive streak formation, the ectoderm get a little bit of functional division. In the middle, you have a slipper-shaped neuroectoderm, and this is the surface ectoderm that forms the skin. So this, when these are the regions, but it is actually the primary primitive streak. From the primitive streak, and another structure forms which differentiate these two. We'll come to that. So what will happen if there is no proper gastrulation? So it will lead to a condition called caudal agenesis. This is the mermaid's deformity, syringomelia, where it will be associated with other anomalies like spinal cord anomalies, cardiac anomalies, and this is syringomelia. If there is, this is premature regression. If the primitive streak disappears early, but if it persists, what will happen? It will lead to sacrococcygeal teratoma. It is a tumor that is seen in neonates, and this tumor will have components developing from ectoderm, mesoderm, and endoderm. It is usually it can be resected also because it has because the primitive streak has pluripotent cells. All can lead to the development of all the germ cell layers. Next. So in this picture, this is the primitive streak. Before that, I forgot to mention about the structure. Wait a minute. So I said that the mesoderm spreads all through, but it is not seen in the midline. Why? Cranially, you have the procaudal plate, which is a close adhesion between the primitive ectoderm and endoderm that persists, isn't it? Later, that forms the oropharyngeal membrane, which ruptures to form the mouth. And also, caudal to the primitive streak, you have the cloacal membrane. And in the midline, there is another structure coming up from the primitive streak that is called the notochord. So these are the areas where you don't have the mesoderm. But cranial to the oropharynx, you can see that there is a mesoderm. That is the area where you have the cardiogenic area, where the heart and the pericardium develops. Okay. So this is the primitive streak, region of primitive groove and primitive streak, and this is the primitive pit. And now we have to talk about the notochord genesis, formation of notochord. That is notogenesis. From the primitive pit, cells migrate cranially in the midline towards the procaudal plate. So what will happen when the cells migrate, as the can see in this picture, as the cells migrate into the region of procaudal plate, the pit also enters it. So the first cells enter the primitive pit and goes cranially in the midline. That is the notochordal process. When the pit extends up cranially, it forms the notochordal canal. So this picture is craniocaudal. You can see the connecting stalk at the caudal end. So in this picture, what is happening? The pit is extending cranially, and at the same time, the floor of the pit and the endoderms come closer together, and there will be openings that develops in this floor. So that finally this floor will disappear. This floor will disappear. As a result, this endoderm and the roof of the notochordal process will be in same line. Getting it? So what is the effect? Here dorsally you have the amniotic cavity, where you have the primitive pit, and because the floor has disappeared, there is a canal that connects the yolk sac with the amniotic cavity. That layer is called the neuroenteric canal. So this is a transit canal which allows the nutrients to pass and gives supply to the dorsal layer, that is the ectoderm. Neuroenteric canal. So that was a craniocaudal cut picture. Now next, what are you seeing? It is a section passing like this, side to side. So what are you seeing? This is the midline, and on the sides you have the intraembryonic mesoderm, and the notochordal floor has disappeared. It is continuous with the ectoderm. And next what happens, and this will form a straight flat plate. Later the plate will curve inwards and multiply and get separated from the surface, and the endoderm will be continuous. This, after it is within the between the ectoderm and endoderm, it forms the notochord, the proper notochord. So this is the process of notochord formation. So what is the function of notochord? You can see the surface ectoderm. It is getting a little bit groove. That midline structure, it's notochord induce the surface ectoderm, the dorsal ectoderm to form the neuroectoderm, just dorsal to it. On the sides, you have the surface ectoderm. So, but that event occurs in the next week. So, this is again craniocaudal picture with the floor disappearing and the primitive pit continuing as the neuroenteric canal. This is the cut. Section B is the cut passing through this, where you don't have the floor. Then notochordal plate is formed. And this cut C is where the notochord is formed, because all this event is in a craniocaudal direction. The disappearance and appearance of the floor and the notochord, it is in the craniocaudal direction. Again, another picture. This is the amniotic cavity. This is the yolk sac, and this red area, it is this part, it is the extraembryonic mesoderm. This part is the extraembryonic mesoderm, but this part is intraembryonic mesoderm. This part, that the extraembryonic mesoderm that lines the amniotic cavity is somatopleural extraembryonic mesoderm. This part which lines the yolk sac is the splanchnopleural mesoderm. So you can see at the periphery of the embryonic disc, the intraembryonic mesoderm is continuous with the extraembryonic mesoderm that lines the amniotic cavity as well as the yolk sac. So what will happen to the notochord? It induces its primitive streak is the primary organizer. It induces the formation of the 3 germ layers. Then it induces the formation of notochord. The notochord induces the formation of the neuroectoderm, which later forms the central nervous system. And but this notochord will disappear. And the remnant that is seen is the nucleus pulposus of invertebrate disc. So you can see the invertebrate disc, which has a fibrous ring in the periphery and a jelly-like substance in the middle. That nucleus pulposus is the remnant of notochord. And the notochord, even though that doesn't contribute to the formation of vertebral canal, they form a framework for it. This is the yolk sac. From the dorsal aspect of the yolk sac, you can see that there is an extension of the yolk sac into the connecting stalk. That is called the allantois or allantoenteric diverticulum or urachus. That in human, it is partly they form to contribute to the development of urinary bladder, and partly they form a remnant that is seen on the posterior aspect of the anterior abdominal wall. So that is it. So in the second week here, we have seen so many events of 2. In the third week, we have seen the 3 layers of the 3 germ layers developing, and the formation of primitive streak, the notochord, and the extension of the yolk sac into the connecting stalk as allantois or urachus. So that is it.
Thank you, ma'am, for delivering such an informative session. Your deep knowledge and ability to simplify complex concepts in embryology have truly enriched our understanding. We now open the floor for questions. Delegates, you are welcome to ask your doubts regarding the topic by unmuting your microphone or typing your questions into the chat box freely. We are encouraging everyone to make this an interactive, enriching discussion. Any more queries? Ma'am, there's a question from Harsha Unni. What's the significance of trophoblastic shell? Could you please
Okay.
explain, ma'am?
Okay, we'll the if the blood vessels and everything go beyond a limit, it will go into the actually the implantation is occurring in the inner two stratum of the endometrium. So that has to be limited by certain factors, no? Otherwise, it will go in deeper and deeper. So that is one function of that. To limit the penetration.
Thank you, ma'am. Any more queries you can freely ask. Ma'am, I think it seems there are no further questions at the moment. I hope all queries have been addressed.
So what happens is when we talk about the intraembryonic mesoderm and extraembryonic mesoderm, don't get confused. The same thing is going to happen in the intraembryonic mesoderm, the two division with similar names. So extraembryonic mesoderm is different. The intraembryonic mesoderm is different, even though it is continuous through the periphery of the embryonic disc.
Okay, ma'am.
So can we stop now?
Yeah, ma'am. I think all the queries have been addressed since. On behalf of everyone here, I extend our sincere thanks to Dr. Geetha ma'am for sharing their expertise and valuable insights with us today. Thank you, ma'am. Thank you once again.
Thank you. Thank you.
Have a great day to all. Thank you.
Thank you. Thank you.